Okay, so let's get started then. Oh, thanks for sharing the link in the WeChat, whoever did that. Hopefully everyone's able to join.
Mr. Shafin,
Yes.
can you upload the note on the WeChat group chat?
Yeah, so are you talking about last week's notes or this week's notes?
Both.
Yeah, so I will upload the notes for a week. The weekend, like 1 week later. Okay, so like last week's notes, you will get them at the end of this week. So like tomorrow. Okay, and then this week's notes, like for this week, you will get them like next week on like Thursday or Friday, I guess, for you guys. Um, so wait, so are you? Would it be helpful if you received it before the quiz, or like is that what you want, or?
Yeah, it would be helpful to see before the quiz.
Okay, all right then. Okay, so in that case, I will send. So last week's notes, I will give them tomorrow, but this week's notes, I will give them before the quiz next week. Okay,
Wait,
in that
when
case.
is the quiz?
So the quiz will be next for you next Friday in
Okay.
your time zone. Yeah. I think yeah. So for me, it's Thursday. It's Thursday night right now. For you guys, it's like what time is it? Like Friday noon, like Friday 12 o'clock right now, or what? Okay. In any case, I think let's get started. So last time we were talking about the endoplasmic reticulum. We talked about the smooth and the rough endoplasmic reticulum. Um, yeah, you're right. Actually, this is a lot of since this is a lot of notes, I will send it to you guys a little bit earlier before the quiz then. I think that will be helpful since it's a lot of things to remember. Um, yeah, so let me go over today the next step in the process, which is called the Golgi apparatus or the Golgi body. And then we'll talk about lysosomes. But in between that, we'll talk a little bit more about the processing of proteins and how it fits into the um, central dogma of biology, like we talked about in the first week. Okay, so to start talking about the Golgi apparatus, I will go back a little bit to um, the endoplasmic reticulum and to this picture, where remember when the proteins get uh, produced into the rough ER, right? They're modified first into their correct shape, like here, and then they're packaged into a vesicle, right? So the question remains is this vesicle does not go straight to its destination unless that destination is the Golgi apparatus, which is another organelle. So first, this vesicle goes to the Golgi apparatus, another organelle, and then it will go to another place depending on if it's supposed to end up somewhere else. So some proteins work in the nucleus, some proteins work outside of the cell, some proteins work in the mitochondria, some proteins work in other organelles, and some just free floating in the cell. And in order to get to their destination, they first are sent by this vesicle into the Golgi body or the Golgi apparatus, and the not the protein, but the whole vesicle is sorted, packaged, and tagged. And the tag that is attached to it lets the cell know where this vesicle is supposed to end up, and that's where it takes the protein. Okay, so to start it off, I'll just write the spelling of it. It's Golgi Apparatus. Sometimes it's called Golgi Body, and this structure is the shape of it is kind of like flat discs, like CDs, and it wraps around the endoplasmic reticulum a little bit. Okay, and it surrounds. It's outside of the endoplasmic reticulum. It kind of surrounds it like a cloth, I guess. Um, so I'll just write the main note that you need to know for this is that the vesicles from ER, meaning endoplasmic reticulum, arrive here, and then they are sorted, packaged, and tagged. So this is the important one. They're tagged. That is the main one I want you to remember. They're tagged so that the proteins or lipids inside them and end up in the right place. The right place. Okay. Now let me just show a picture because the anatomy of this is a little bit important for understanding the mechanism, the central dogma mechanism, which I will explain in just a second. So here we have the nucleus right here, right? And here we have the rough ER. So smooth ER is also somewhere. It's just not in this picture, right? And you see there's little orange dots. These are the ribosomes. Is this picture big enough for everybody to see? Okay.
Yes, I think this side is okay.
Okay, so these little dots are ribosomes. This is the ER, specifically the rough ER, although it says right here. So I won't write that. Keep in mind, most cells will also have the smooth ER. It's just this picture does not show it. Right, and then this is the Golgi apparatus. You can call it apparatus or you can call it body. I don't care. Now, this right here is one of the vesicles that I was talking about. It's like a little bubble. Okay, it's a little bubble that carries a protein. Okay, now what ends up happening is when the vesicle leaves the ER, it can have a tag, but that tag is a carbohydrate which tells the vesicle to go to the Golgi apparatus. Okay, and then if it doesn't have that tag, so not all of the vesicles from the ER will have that carbohydrate tag. If it doesn't have it, then the protein will not go to the Golgi apparatus. Okay, they will go straight to the cytoplasm, and then it will operate in the cytoplasm because that is, where those proteins are supposed to operate. But if it's supposed to operate anywhere other than the free floating in the cell, it will have a Golgi apparatus tag so that the vesicle ends up in the Golgi apparatus like this. Okay, see this this green thing right here, that green thing, that is a carbohydrate tag that is attached to this vesicle, and it tells the vesicle to go to the Golgi apparatus. Okay, now once it reaches the Golgi apparatus, the tag disappears from the vesicle, but it stays with the protein like this. Okay, but it will undergo further modification that tells the cell specifically where it's supposed to go, not just the Golgi apparatus. The Golgi apparatus is just a transportation hub, kind of, and it will add different tags. So it has this Golgi tag which stays on, but the Golgi apparatus might add another one, such as see plasma membrane, or it might add one for I don't know, something else, something different, for the nucleus maybe, or for the mitochondria. So if I had to draw out the whole diagram of exactly what's happening in the life cycle of a protein, and I want you guys to try to take note of this as best as you can. So this is, again, I will draw the central dogma of biology, but I will go a little bit further than the central dogma. So we have DNA, right? This is in the nucleus. Then we have the mRNA, or you can just call it RNA, whatever. And this is free floating in the cytoplasm. Okay, and then we have the protein. This is also either free floating. So it's either free floating and then it goes into the ER, okay? Or it's made straight into the ER, okay? So proteins can either be made to be free floating and then go to the ER, or they can just go straight into the ER, okay? Or be made straight into the ER. And also some free floating proteins also just, that is the end of their processing. That's just how they are, okay? They can process themselves because they're small and simple. So from the ER, there's two options from the ER. We're still working with the proteins, right? So the protein can go to two places. So one is free floating again. Free floating. So in the ER, what happens is protein processing. I'll just write it that way. And after the protein is finished processing, and by processing, you remember I just mean it folds properly, it has attachments like carbohydrates if it needs to be added or whatever, anything, right? So after the processing, if there's no tag, so no carbohydrate destination tag, no destination tag, so no destination tag, then it becomes free floating. Okay. The other option is it has a tag, so destination tag, then it goes into the Golgi apparatus. So depending on the specific type of tag that is added there, the Golgi apparatus will analyze this tag, and further modifies. Protein. And then from there, it can go to the nucleus. This has a nuclear tag. Or it can go to the plasma membrane. That's if it has an export tag. Or it can go to the mitochondria. Again, if it has a mitochondria tag, I'll just call it the MT tag. And then it can also go to any number of other places. So I'll just say it's etc. Okay, so does this processing system make sense? I know it's a bit confusing and it's probably a lot to take in, but
I have a question.
Yes.
What kind of tag does the mitochondria have?
You mean specifically, or?
Yeah, I want to know the name.
So I don't. So, okay, so when I call it these tags like nuclear tag, export tag, mitochondria tag, etc., I'm referring to a category. Okay, there is not one single nucleus tag or one single mitochondria tag. Okay, for our purposes, since we're not in a biochemistry class, we're just in a biology class. We just have to know the category. So all you need to know is that if there's a general mitochondria tag, it will go to the mitochondria. Okay.
Okay.
If you want to know specific names, I I'm trying to remember from when I took biochemistry, but I don't remember the mitochondria ones. There's one I remember from the plasma membrane. There's two that I remember, but I'm trying to remember the names. Yeah, I can't really. The names are really kind of like they're technical and they're hard to remember, so I'm not gonna test you on them. But there's one for the plasma membrane. It is called CLK1 and CLK2. CLK1 takes it straight to the plasma membrane. CLK2, the tag, it takes it to the plasma membrane and then outside the cell. Okay, but CLK1 causes it to stay attached at the membrane.
Mr. Malik,
Yeah.
it is important for our quiz.
The names?
Yeah.
No. I just want to make clear. This stuff you do not need to. I'm only saying this because he asked for an example. Okay.
All right, thanks.
And then another one he asked for an example was there's one called COP2. So COP2 is another example. And then for the nucleus, I think there's one called COP1 is a tag. I don't remember any of the names for the mitochondria tags. So none of these things I just wrote in red right now, you don't need to know any of them. All you need to know is that the only thing you need to know is that in the Golgi apparatus, whatever tag there is, it will be understood by the Golgi apparatus, and then depending on the type of tag, it will go to the correct location, assuming everything in the cell is working correctly. Okay? So if the Golgi apparatus reads that there is a nuclear tag, it will send the vesicle to the nucleus. Okay? Does that make sense to everybody? Any other questions? Okay. By the way, somebody told me last week, what time does the class end on Fridays for you guys?
So the event is over, so you can just end the class normally.
Oh, okay, that was for an event. Okay, I understand.
Okay.
So the class will end at a normal time today?
Yes.
Okay, all right, that's fine. It's weird because the school did not tell me about that, I think. So I just didn't know. All right, that's fine. So the next thing we will talk about, right, one more thing about the Golgi apparatus so I'm assuming everybody has this chart understood at a basic level at least. Okay, so I'll go back to the Golgi apparatus a little bit. One more thing I do want you guys to remember for the quiz. So you'll notice here this word right here. It says cis face right there. Do you see it? And then trans face. So in the Golgi apparatus, so cis face is the Golgi side that faces the ER. Okay? And it's the receiving side. And then the trans face faces away from ER. Then this is the export side. Maybe I make this a different color so it's easier to read. Okay. I'll let you guys copy that for a second. Okay, now just one final thing about the Golgi apparatus. So because the Golgi apparatus is used in export and basically the export of proteins, it follows that cells that export more proteins will have more Golgi apparatus stacks. So what this means is that they will either have more Golgi apparatus like in number, or it will be much bigger. Okay? So some examples of the cells that make a lot of Golgi apparatus, and I'd like you guys to remember just two of these examples. So one of them is your immune cells, which produce antibodies, produce antibody proteins. And saliva, sorry, I should call it salivary gland cells produce saliva in mouth. And then third one is chief cells or stomach cells, stomach acid cells. Stomach acid cells produce acid resistant proteins. Okay, so please try to remember at least two or at least one of these examples. I don't know yet, but I might ask in the quiz for an example of a cell that produces more proteins. If you do research on your own and you find another example, that is also fine. Let me try to think of a few more. I can't quite remember. So another example would be muscle cells. Muscle cells. Export proteins and other molecules when exhausted. Okay, so that's another example. Okay, any questions about this before I move on to the next organelle? Okay. All right, so one of the important but smaller organelles in the cell that we will talk about is called lysosomes. And lysosome is also a membrane-bound organelle, so it has its own plasma membrane inside of the cell. Okay. So this is, if you want to think of it in the simplest term, is the recycling factory of the cell. It is usually acidic inside. Breaks down anything that enters. Okay. So this process that I just described, so in every cell, this will happen for all old structures. Okay, so this is usually happens to old structures that need to be rebuilt. rebuilt. So what will happen is that, for example, suppose there is a protein, right? Suppose there is a protein that is old. It is starting to break down, right? What will happen is that that protein will go to the lysosome, and then it will be broken down into its components, either atoms or amino acids, which are the monomers, remember? And then those monomers will then be available in the cell for other reactions to happen so that proteins can continue to be built. So it's basically recycling usable components. Okay? Now, lysosomes are in almost every cell. But there's one type of cell in particular, or one category of cell in particular, where the lysosomes are very, very important. So they're a category of white blood cells actually two categories, not just one of them. So not all white blood cells. I'm not talking about all white blood cells for now. What I'm talking about is two categories. One of them is granulocytes, and the other one is macrophages. Okay? So in macrophages, they're just lysosomes. Many lysosomes. Somes that are very acidic. Very acidic. But in granulocytes, the lysosomes are called granules. Okay? And or at least some of the lysosomes are called granules. And these lysosomes, granules, specifically only break down pathogens, which are harmful bacteria and viruses. Okay? So in white blood cells, there are many, many different types of white blood cells, at least 30 different types. So I'm talking about two categories of them. So this is a category of its own, and it's one type of cell. This is another category that contains three types of cells. So these two categories together, which is makes two categories is four different types of cells. They have lysosomes which break down pathogens and viruses, basically. Okay. Any confusion about this? Okay, so let me demonstrate with the picture really quickly what happens here in the white blood cell. So this picture right here is not for granulocyte. It's showing a macrophage. Okay, but what happens in the granulocyte is almost the same thing. So it's a pretty good representation. So what this picture is representing a macrophage type of white blood cell. Okay, and what will happen is that this pathogen, just pretend this is a bacteria. Okay, we'll just call it a bacteria or virus. Now that bacteria will enter the cell. It kind of like a what's the word? I just forgot the word. A vesicle. A vesicle. Remember how the protein will leave the Golgi apparatus in a vesicle? So just like that, when the bacteria is eaten, the cell eats the bacteria. It will enter kind of a very large vesicle, which we usually call the phagosome, like this. Okay, Phagosome is just a vesicle. It just specifically means a vesicle that has bacteria or virus inside. Okay. Now, these are the lysosomes inside the white blood cell, which will be used to break down the bacteria. So if this was a granulocyte instead of a macrophage, it would say specifically it would say granules instead of lysosome. But either way, the same thing would happen. This lysosome will combine with the phagosome, which has the bacteria, and will form one large vesicle. And then the digestive enzymes at the acidic environment will break down the bacteria into millions of pieces, and it's dead at that point. It is completely dead. And then the cell will just send it back out, the waste products. Okay, and just like that, this white blood cell killed a bacteria, a harmful bacteria, during an infection. Okay. Any questions about that?
I have a question.
Yes.
Is phagosome also a lysosome, and the one that has the digestive enzyme also a lysosome?
No. So a phagosome is not an organelle or anything. It's just a container for when the white blood cell eats the bacteria, it makes so any type of transportation into or out of a membrane creates a vesicle. Okay, so when the cell eats the bacteria, it creates a sphere around it, which is a vesicle. So that vesicle that has the bacteria in it, we just specifically call it the phagosome. Okay. Now the phagosome is not a lysosome. It's just a special vesicle that carries the bacteria. The phagosome will combine with a lysosome to create a large um, you can call it a large lysosome, I guess, at that point. At that point you can, when it combines with the lysosome, you can call it like a large lysosome with all the digestive enzymes and acids. Okay? Which then kills the bacteria and then it sends it back out. Does that answer the question?
Yes, I think it makes sense.
Okay, cool. If anybody else has any questions, just feel free to ask at any time. Okay, so that's about it for lysosomes. We have about 3 minutes left. I will talk about. I'll see if I can talk about this in 3 minutes. So the next organelle we will talk about is vacuole. So vacuole is it is similar to a lysosome. A lysosome. Only in plant cells. And this is extremely important. The vacuole is only in plant cells. Okay. And this is a large central organelle. Which sometimes, some cases, can cover more than 30% of cell volume, or the cell's like space. Okay. In addition to working like a lysosome, so other than tasks similar to lysosomes, the vacuole also stores water and waste. So stores water and waste and isolates harmful substances. and I will try to. I did not prepare a picture for this. I didn't think we would get this far today, but before I let you guys go, let me just show a picture, so that you have an idea of how large it is and what it looks like. So, hopefully I can find a good picture. Okay. So this is a picture of a plant cell. It looks very different from an animal cell, but inside it's actually pretty similar, except for a few exceptions that we will talk about next class. But here you see the vacuole. So in this plant cell, the vacuole is actually a little bit smaller than in some other plant cells. It's maybe like 10% or 15% of the total area, the total volume. In some plant cells, maybe it can go up to like 40% of the whole size. Okay, and this is basically what it looks like. All right. Yeah, I think that is the end of class. So if anybody has any questions, you can ask me. I'll stay for a few more minutes, and if you don't, then you're free to leave.